Pleasure and honor of introducing our speaker this morning, Toshiyuki Shimizu. Professor Shimizu graduated in 1989 from the University of Tokyo. From 1989 to 1995, he worked at Kirin Brewing Company and also worked as a researcher at the Protein Engineering Research Institute in Osaka. From 1995 to 2001, he was a research associate at the Nara Institute of Science and Technology. From 2001 to 2010, he was associate professor at Yokohama City University. Since then, he's been professor in the Graduate School of Pharmaceutical Sciences at the University of Tokyo. Professor Shimizu's research group has determined a wide variety of structures. Looking at the PDB, I found 170 deposits from his group. The most recent, guanine nucleotide exchange factor, SMG-GDS, which has beautiful armadillo repeat motifs. Other major recent crystal structures this year, structure of human Izumo-1, Juno-1, and the complex between Izumo-1 and Juno involved in control of fertility. Professor Shimizu's extensive work on the innate immune system has focused on the toll-like receptors. His lab has determined structures of TLR7, TLR8, TLR9, and complexes of TLR4 with MD2. His group has also determined the structure of the NOD-like receptor, NOD2. And today, Professor Shimizu is going to talk about his TLR work with a lecture on structural studies of single-stranded RNA sensing TLRs in the innate immune system. Professor Shimizu.
This? Oh, sorry. Sorry. Thank you for your kind introduction. I'm Toshiyuki Shimizu from the University of Tokyo, Japan. It is a great pleasure for me to have a talk in the IUCR Congress as a keynote lecture. Today, I'd like to talk about toll-like receptor sensing a single-strand RNA working in an innate immune system. We are surrounded by a wide variety of pathogens such as viruses, bacteria, and microorganisms. We are always in danger of the infection of these pathogens. To cope with this, we have a system for eliminating them to protect ourselves. This is immunity. When pathogen enters the body, we initially attempt to eliminate it by innate immunity. Toll-like receptors, TLR, sense the invasion of pathogen by recognizing pathogen-associated molecular patterns such as a lipid, a protein, and a nucleic acid derived from the pathogens. And then inflammatory reaction is activated. One of the most important roles of innate immunity is to activate adaptive immunity. Dendritic cells are bridging innate immunity and adaptive immunity. After the activation of adaptive immunity, T cells and antibodies attack the pathogens. This slide shows the intersignaling pathway of toll-like receptors. To date, 10 types of TLR have been identified in humans, and each TLR recognizes a specific pump. For example, TLR4, which is the first identified TLR, forms a stable complex with MD2, and this complex recognizes LPS, lipopolysaccharide. And TLR7 and 8 recognize a single-strand RNA. And the localization of TLR is different. TLR 1, 2, 4, 5, 6 are mainly expressed on the plasma membrane, and the TLR 3, 7, 8, 9 reside in the intercellular organelle and lysosome. TLR is a type 1 integral membrane protein and consists of 3 domains: extracellular domain, transmembrane domain, and the intercellular domain. The intercellular domain is known as the TLR domain, which plays an important role for signal transduction. And extracellular domain is a ligand binding domain and contains tandem copies of leucine rich repeat motifs. And the number of copies are different among TLR. This slide shows the activation mechanism of TLR by ligand binding. Generally, TLR exists as a monomer in a unligandic form. And binding of agonistic ligand causes dimerization of extracellular domains, leading to the association of TLR domain. And this triggers the recruitment of specific adapter proteins such as MIT88 and the TRAM to associate it to TLR domain, initiating a downstream signaling cascade. To date, several crystal structures of extracellular domain of TLR have been reported. All structure domain display a horseshoe structure, and ligand binding induces dimerization, giving rise to M-shaped structure, M-shape, and ligand protein and the protein-protein interaction brings the C-terminal region of extracellular domain into close proximity. Based on the crystal structure of TLR3 complex with a double-strand RNA, the dimerization model of full-length TLR has been proposed. TLR 7, 8, 9 are highly homologous and forms a subfamily called TLR7 family. This family recognizes a single-stranded nucleic acid. TLR7 and 8 recognize a single-stranded RNA, and the TLR9 recognize a single-stranded DNA containing a CPG motif. And this family has a long insertion loop called Z loop. And the cleavage of Z loop is necessary for the activation. TLR7 family exhibits a more complicated activation mechanism. Before the cleavage of Z loop, TLR7 family exists as an inactive monomer. And after Z loop processing, TLR7 family was transformed into an inactive dimer. And after the ligand binding, TLR7 family forms an active dimer. TLR7 and 8 recognize a single-strand RNA, and interestingly, these proteins are also activated by small chemical ligands such as imidazolquinoline, thiazolquinoline, and guanosine derivatives. These chemical structures of these compounds similar to the base of RNA. And especially RH37 imiquimod is approved for the certain diseases, such as genital warts. So the question we have, how do these ligands activate the TLR8? To answer the question, we started the structure study of TLR8. A couple of years ago, we saw the crystal structure of human TLR8 in unliganded form and small chemical ligand form. This is the first crystal structure of TLR recognizing a single-stranded nucleic acid. And TLR8 displays a more circular structure, not the horseshoe structure. And our structure work identifies a ligand binding site and provided a detailed recognition mechanism. The C-termini are brought into closer proximity upon the ligand binding, which is appropriate for the association of TIR domain. And compared with the two structures, ligand binding in this large structure reionization. This is a side view of the TLR8. The ligand binding induces a large conformational change accompanied with ring rotation and hinge motion. This is a movie. This is a side view of the TLR8, and this is an unliganded form. And ligand binding induces a large conformational change, and the C-terminal brought into closer proximity, which are appropriate for the TIR association. As I mentioned, TLR8 and 7 recognize a single-strand RNA, and small chemical ligand is similar to the base of RNA. But RNA molecule itself is quite different from small chemical ligand because RNA is a large molecule linked by a phosphodiester bond and heavily negatively charged. This is an example of agonistic RNA of TLR8. TLR8 prefers a UG-rich sequence. So the question we have, how does TLR8 recognize a natural ligand single-strand RNA? And how and why can different ligands, RNA and small chemical ligand, similarly activate TLR8? To answer the question, we started a structure study. This is a crystal structure of TLR8 in complex with a single-strand RNA. For the crystallization, we use RNA consisting of 20 bases, but we cannot observe the electron density corresponding to intact RNA. Instead, we observe the reading and the short oligonucleotide UG. So TLR8 recognizes reading and the short oligonucleotide UG at two distinct sites. The binding site of reading here is equivalent to that of a small chemical ligand. We call this site the first site. And the short oligonucleotide found a newly identified site on the concave surface of TLR8. We call this site second site. We did not observe the intact RNA. To explain this, there are two possibilities. One is observed molecules or bona fide degradation product. The other possibility is that parts of intact RNA were observed and other parts were disordered. To confirm this, we conducted the LCMS analysis of dissolved crystal. This is the result. So we did not detect intact RNA at all. If crystal contains intact RNA, the peak should appear at this position. And reading mononucleoside was the most abundant species. And two short oligonucleotide, UG and UUG, were also detected. So this result strongly suggests that observed molecule, reading and UG, were degradation products derived from single-strand RNA. And UG at the second position would be a mixture of UG and UUG. This is a close-up view of the ligand binding site, first site and the second site. Reading bound the first site on the dimerization interface, bridging two protomers. So, uridine acts as a molecular glue, bridging two protomers. And short oligonucleotide bound the second site on the concave surface of TLR8. And this short oligonucleotide is not involved in the dimerization interface. Uridine and the small chemical ligand is recognized in a similar manner. There are three key interactions for the recognition. One is a stacking interaction with phenylalanine, here and here. This interaction is conserved between two structures. And second is a hydrogen bond with aspartic acid. This hydrogen bond is also conserved, but in the uridine complex, partially collapsed. Instead, water-mediated interaction is observed. Most remarkable feature is hydrophobic interaction. Chemical ligand has a long alkyl chain, and this alkyl chain is snugly fit into a hydrophobic pocket created by two protomers. But uridine does not have such an alkyl chain, so accordingly, this pocket is empty. So accordingly, binding affinity of uridine is much weaker than the chemical ligand, as I mentioned after. To confirm the functional importance of the residues at the first site and the second site, we mutated these residues and examined the ability of these mutants by NF-kappa B assay. Mutation of the first site residues abolished and reduced response to a small chemical ligand and single-strand RNA. And mutation of second site residue also reduced the response to a single-strand RNA, but response to a small chemical ligand does not change. This result is well consistent with the structural work. Small chemical ligand only bound to first site, but the single-strand RNA bind to first and the second site. The first site is essential for the recognition of both ligands, but the second site is important for a single-strand RNA, but not for a chemical ligand. We directly measure the binding affinity for mononucleoside. We tested uridine, histidine, T, A, G, 5' UMP, and 3' UMP. Among them, uridine had the highest affinity for TLR8 with a KD value of 55 micromolar. But this value is very weak compared with chemical ligand whose dissociation constant is 0.2 micromolar. To overcome the discrepancy, some other factors should be considered. So our structural work suggests that oligonucleotide binding to the second site influence uridine binding to the first site. So we conducted the ITC analysis again in the presence of oligonucleotide. So this is the result. The binding affinity for uridine in the presence of oligonucleotide was significantly enhanced to increase to 1 micromolar, which is comparable to the small chemical ligand. And we also performed the NF-kappa assay at the low concentration of single-strand RNA where no activation was observed. In this condition, addition of uridine significantly enhanced the activity. So uridine and oligonucleotides synergistically activate the TLR8. So TLR8 is a single-strand RNA receptor. It's true, but TLR8, our structural work suggests that the TLR8 is a uridine sensor in the presence of oligonucleotide. This is a summary of my first topic. We determine the crystal structure of TLR8, unliganded form, chemical ligand bound form, and a single-stranded RNA bound form. And ligand binding induces a reorganization of the TLR8 dimer. And the TLR8 is a sensor for uridine and oligonucleotide derived from RNA. And the TLR8 exhibits a synergistic effect for uridine and oligonucleotide. And the chemical ligand exhibits a sufficiently high affinity to activate the TLR8 by themselves. As I mentioned, TLR7 family requires Z-loop processing. And several proteins are identified to cleave the Z-loop. For example, furin-like competitors, cathepsin, asparagine, and peptides. And why Z-loop cleavage is necessary for the activation? So to review the Z-loop, the functional role of Z-loop, we started the structure study. Wild-type TLR8 is automatically cleaved during the purification. So to avoid proteolytic cleavage, we mutated cleavage site with other sequence. So so we can successfully purify the Z-loop mutant. This is a SDS page. Wild-type TLR8 shows a 2-band because proteolytic cleavage. But Z-loop mutant shows one bond. We also characterize a Z-loop mutant. This is a gel filtration chromatography, and wild-type TLR8 is eluted as a dimer in the absence or presence of the agonistic ligand. But Z-loop mutant is eluted as a dimer, eluted as a monomer, irrespective of the agonistic ligand. And we also perform the NF-kappa B assay, and wild-type TLR8, of course, respond to agonistic ligand, but Z-loop mutant does not respond to agonistic ligand. So Z-loop mutant is inactive, irrespective of the agonistic ligand. After the screening, we obtain a crystal of Z-loop mutant. This is a crystal structure of Z-loop mutant. In the crystal structure of the wild type, the N-terminal part of Z-loop is disordered. So, TLR8 has a two lateral phase, ascending lateral phase and a descending lateral phase. So, in the previous work, we cannot determine which lateral phase Z-loop passed. One is here or here. So we cannot determine which is right. But our structure of Z-loop mutant clearly shows that the Z-loop passed the ascending lateral phase. This region corresponding to this region. So Z-loop passed the ascending lateral phase. So this structure is superimposed to the dimeric structure. Z-loop is heavily crashed with the binding partner. So this work clearly demonstrates the functional role of Z-loop. The uncleaved Z-loop prevents the dimerization of TLR8 by steric hindrance. And TLR7 family has a similar Z-loop. So this mechanism would be occurring in TLR7 family. This is a summary of my second topic. We determined the crystal structure of Z-loop mutant, and we show that functional role of Z-loop. As I mentioned, TLR7 and 8 is highly homologous and recognizes a single-strand RNA. But the expression pattern of these proteins are different. TLR8 is expressed in a macrophage and a myeloid dendritic cell. In contrast, TLR7 is strongly expressed in the plasmacytoid dendritic cells. So expression pattern is quite different. But are there any other difference between the TLR7 and 8 from the biochemical and the structural viewpoint? As I mentioned, TLR8 is activated by reading in the presence of single-strand RNA. Our collaborator, Professor Miyake and Shibata's group, show that TLR7 is activated by a guanosine in the presence of a poly-U. And we also conducted the SVAUC analysis. So small chemical ligand alone can dimerize the TL7, but poly-U and guanosine does not induce the dimerization. And poly-U and guanosine synergistically triggers the TL7 dimerization. So considering these results, TL7 and 8 shows, of course, similarity, synergistic effect, but ligand preference is different. So to reveal the similarity and the difference of these two proteins, we started the structure study of TL7. It is necessary to obtain the sufficient amount of purified protein for the structure study. We check the expression level and its properties of TL7 from various species. Among them, a monkey and a pig TL7 shows a good property. And zero processing is required for the activation, so we introduced a thrombin site into a Z-loop region. And Z-loop processed protein was prepared by a thrombin digestion. After screening, we successfully obtained crystal of TL7 complex with guanosine and poly-U, and the TL7 complex with a small chemical ligand. This is a crystal structure of TL7 complex with guanosine and poly-U. For the crystallization, we use a poly-U consisting of 19 bases. TL7 recognizes guanosine and short oligonucleotide UUU at two distinct sites. The binding site of guanosine here is equivalent to that of reading in TLR8. So the first site is the same. But the oligonucleotide binding site are different in TL7 and 8. So second site is completely different. Guanosine and small chemical ligand are recognized in a similar manner. Similar to TLR8, three key interactions are important for the recognition. Stacking interaction with the phenylalanine and the hydrogen bond with the aspartic acid, and small chemical ligand has a long alkyl chain, and this alkyl chain is snugly fit into a hydrophobic pocket, but in the guanosine structure, hydrophobic pocket is empty because guanosine does not have such a long alkyl chain. And this is a close-up view of the ligand binding site at the second site. At the second site, we observe the UUU oligonucleotide, but according to the result of LCMS analysis, this poly-U would be a mixture of various species. Most abundant species is a tetra-U. UUUU is the most abundant species, but we observe the UUU oligonucleotide. And the middle U, U2, is strictly recognized by multiple interactions with TL7. But the flanking bases U1 and U3 is loosely recognized. And 2' hydroxyl group, which is specific for RNA, is also recognized TL7. This suggests that TL7 RNA can bind to a TL7, but DNA does not bind to TL7. To confirm this, we perform the gel filtration analysis. This is the result. Poly-U can bind to a TL7, but poly-DU DNA does not bind to TL7. To confirm the functional importance of the residues, first and the second site, mutation of the first site residues abolished the response to a small chemical ligand and a single-strand RNA. And mutation of the second site residues reduced or abolished the response to a single-strand RNA, but response to a small chemical ligand does not change so much. So the first site is essential for recognition of both ligand. And the second site is important for single-strand RNA, but not for a chemical ligand. To further confirm the importance of the residues at the second site, we examine the binding and the dimerization ability of these mutants. This is a result. Second site mutant shows reduced binding to poly-U here, but small chemical ligand bind to TLR8, second site mutants of TLR7. And small chemical ligand can induce a dimerization of second site mutants, but poly-U cannot induce the dimerization. We directly measure the binding affinity for mononucleoside. We tested the guanosine, uridine, A, T, C, I, xanthosine. Among them, TLR7 exhibits strong affinity for guanosine, but dissociation constant is weak compared with a small chemical ligand. So, considering the result of TLR8, we conducted the ITC analysis again in the presence of poly-U. Expectedly, binding affinity is significantly enhanced in the presence of poly-U. And small chemical ligand, binding affinity, small chemical ligand is also increased in the presence of poly-U. And we examine the sequence specificity of single-strand RNA at the second site. So far, we use poly-U as a single-strand RNA. So, in addition to a poly-U, we tested the AAA, GGG, CCC, UCU, and UGU, and examined the dissociation constant. The synergistic effect was observed in the presence of UUU, but the other RNA does not show a synergistic effect. So we concluded a non-terminal reading in RNA is necessary for the synergistic effect on TL7. This result is well consistent with the structural work because middle U is strictly recognized, but flanking base is loosely recognized. So this structural feature is well consistent with these results. This is a summary of my third topic, single-strand RNA containing a non-terminal reading bind to the second site of TL7. And single-strand RNA loaded TL7 has a higher affinity for guanosine, synergistically forming an activated dimer. And the small chemical ligand can directly activate the TL7 without any binding of single-strand RNA to the second site. We determine the crystal structure of TLR8 and the TL7. Both proteins are highly homologous and shows, of course, similarity, but have certain differences. TLR8 and the TL7 has two binding sites, first site and the second site. First site is a mononucleoside and a small chemical ligand binding site. And the location of the first site is the same. But the TLR8 accommodates reading, and in contrast, TL7 accommodates guanosine. And the second site is a single-strand RNA binding site. TLR8 prefers UG-rich sequence, but its sequence specificity is very weak. However, TLR7 requires a single-strand RNA containing a non-terminal reading. And the position of second site is quite completely different. So TL7 and 8 is a single-strand RNA receptor. It's true, but TL8 and 7 is a dual receptor for mononucleoside and oligonucleotide. Especially TLR8 requires a reading, so TLR8 is a reading sensor. Of course, single-strand RNA is necessary. TL7 requires guanosine and a single-strand RNA containing reading. So this TL7 is a dual sensor for these ligands. And we demonstrated, we showed the functional role of Z-loop, and Z-loop prevents dimerization by steric hindrance. Interestingly, TL7, the conformation of TL7 family is quite different. We also determined the crystal structure of TL9 and showed three structures. TL7, the Z-loop of TL7 is here, and the TL8 is here, and the TL9 is here. So Z-loop in TL7 and 8 is involved in the single-strand RNA recognition, but oriented in the reverse direction. And the Z-loop in TL9 is not associated with the ligand recognition. So Z-loop, the conformation and the role of Z-loop are different among the TL7 family. Taken all together, we propose a mechanism of single-stranded RNA sensing TLR. Pathogen and self-derived RNA are delivered to endolysome, and in the endolysome, these ligand, RNA and other ligand, are digested by appropriate enzymes such as ribonuclease, phosphatase, to yield a shorter fragment or mononucleoside. So degradation products are pooled. Uncleaved Z loop sterically hinders the dimerization of 2 TLR. So if TLR7 and 8 is delivered to unfavorable place, so the Z loop cleavage does not occur, so TLR7 and 8 is inactive. But if TLR7 and 8 is correctly delivered to endolysome, the processing is occur, so the inactive dimer was created. So, and degradation product synergistically activates TLR7 and 8. And synthetic ligand, the binding affinity of synthetic ligand is very high, so synthetic ligand can directly activate the TLR7 and 8. So this work is strongly collaborated with Professor Miyake and Shibata's group, the Institute of Medical Science, the University of Tokyo, and LCMS analysis is performed by Toshiyuki Sobe's group, the Tokyo Metropolitan University, and AUCSV analysis is performed by Susumu Uchiyama's group, Osaka University. This is a photo of my laboratory, and I especially thank Dr. Otto, Ms. Tanji, and Mr. Chan. Thank you for your attention.
Thanks for the beautiful talk. Any questions?
In your nice talk, you have shown that the toll-like receptor interacts with small ligands, but there are cases known where the toll-like receptor is activated by big proteins. Do you think that in that case the interaction is between the entire protein and the toll-like receptor, or do you think it is between the degradation product of the protein and the toll-like receptor?
Case what? So sorry.
Sorry, I said that when the toll-like receptor is activated by big proteins.
Yeah.
Do you think that in that case the interaction is a direct interaction between the protein and the receptor, or is in that case could be a degradation product of the protein?
Our data shows that degradation products directly activate TL7 and 8. So other factors are not related with activation according to our results. So maybe the other factor should be considered, but currently we did not detect the other factor for the activation in the TLR7 and 8.
Other questions?
Do you think there's any other role for the Z loop in, for example, recycling of receptor, preventing its degradation? Because it's not clear that you need a Z loop if you've got an inactive conformation when it's cleaved.
Yeah. Good question. I don't mention the role of Z loop. I focus on the steric hindrance of Z loop, but Z loop itself is the conformation of Z loop is quite different, I mentioned. So interestingly, Z loop is directly is involved in the ligand recognition. So for example, if TL7 has a similar conformation of TLR8, so the bindings, the second binding site is occupied by this Z loop. So Z loop conformation is important for the ligand binding. So the functional role of Z loop, main functional role of Z loop is steric hindrance, but also plays an important role for ligand recognition. So the Z loop is partly participate in the ligand recognition.
Other questions? Yes.
The uridine and guanosine binding, is it irreversible or reversible?
Sorry.
Uridine and guanosine, they bind to the receptor, right? The binding is reversible or irreversible?
Maybe reversible. You mean the guanosine bind to for example, TLR8?
Yeah.
According to the result of ITC analysis, we did not detect guanosine binding in TLR8.
Yeah.
So at very high concentration, guanosine might bind to a TLR8, but it's a normal condition. So in the cell or normal condition, guanosine does not bind to TLR8 in the presence of the single-strand RNA. So our experiment suggests.
And the Z loop processing, is it signal dependent, or every time constitutively it degrades the Z loop?
Yeah, the Z loop regulates what?
Proteases acts on Z loop and further binding of this dimerization of ligand receptor, right?
Yes.
Every time Z loop processing happens in the cell, or is it signal specific?
No, maybe. Once the TL7 and 8 is delivered to an endolysome, an endolysome has various proteases. So probably this cleavage is spontaneously occur. But if, as I mentioned, TL7 and 8 delivered to an unfavorable place, the protease does not, there is no protease, so TL7 and 8 remains inactive, I think.
Okay, thank you.
Okay, let's thank Professor Shimizu again for a wonderful talk and hear a couple words from Ard Banerjee.
On behalf of the organizers, it's a real pleasure to present a token of appreciation to the speaker, Professor Shimizu. Also, on behalf of the organizers, it's a real honor to present a token of appreciation to the chair of this keynote lecture. Thank you very much. Thank you very much.